right. So for those who have watched our previous webinars, I'm Rachel Hostetler. I'm a scientist at the Allen Institute for Brain Science, and my role is to make some of our taxonomies and data sets and tools more accessible to other researchers outside of the Institute. And part of that includes this webinar series where we've been doing webinars with slideshows and presentations, and also office hours to allow for some Q&A and more in-depth discussion with some of our other Allen Institute scientists. And so today I'm joined by Emma Thompson and Aaron Garcia, and I'll let them introduce themselves.
Hi, thank you, Rachel. And hello, everyone. My name is Emma Thomas. I'm a scientist in brain science, particularly on the molecular genetics team. I've been here for about 3 years, and my role is kind of twofold. One is in helping to develop and test new cutting-edge transcriptomic technologies that allow us to generate a lot of the data that we analyze, and then also to analyze a lot of large-scale transcriptomic data for the sake of identifying novel genetic tools that can be used by the broader community.
Nice. I'm Aaron Garcia. I'm a scientist also here at the Allen Institute for Brain Science on our human cell types team, working largely on the human and mammalian brain atlas project, where my role is kind of on both endpoints of data generation, where I'm responsible for doing the majority of the dissections of the actual brain tissue, and then sort of as the data returns, also involved in generating some of the taxonomies of the sequence data, specifically for human and non-human primates primarily.
And I should mention, in contrast to Aaron, my work focuses on mouse.
Yeah, so we have many species represented here in our group, and these are the main areas that the Allen Institute works on. If you've seen any of our taxonomies, it's primarily mouse, non-human primate, or human. And yeah, so anyone can chime in at any point, submit a question in the Q&A box if you have a question. If not, I will start off with some questions that we have about, single cell and single nucleus RNA sequencing. So the first question that we have is, can either of you describe the general steps in the process, starting from, obtaining the tissue all the way through having your data ready for analysis? And what are some common platforms that you use? Perhaps any specifics for any of the platforms, or, maybe you use certain platforms for certain areas. Just, generality, how does this work for someone who perhaps has not done these experiments before?
Yes, I can take an initial stab. But yeah, so the workflow is essentially you start off with some tissue, we'll say brain tissue as the example here. You do some initial quality control, you make sure that the RNA integrity scores are good before you process it. And then depending on whether you're doing single cell or single nucleus, both steps involve various degrees of breaking down or homogenizing that tissue to either free the cells, full intact cells, or free the nuclei. Then we'll typically try to enrich for specific populations at the broadest level. For the HMBA work, we'll do enrichment for neuron populations and try to effectively filter out a lot of the oligodendrocytes, which are pretty promiscuous, especially in primate species. After we do fluorescence activated cell sorting, so enriching using FACS, everything, or the final nuclei will be then loaded onto, typically we use 10x platforms for a lot of our sequencing. So they'll be loaded on some form of a 10x chip. So whether we're doing multiome, that would be the 10x multiome version 3.1 chip. For some projects, we're doing multiplexing, which utilizes 10x's on-chip multiplexing technology. We can get into some of the specifics if folks have questions a little bit later. But effectively, as things are loaded onto the chip, the nuclei are being separated by oil effectively, and then they're also being tagged with a gel bead. And this gel bead kind of provides the necessary barcoding for being able to identify cells downstream. But it also provides a lot of, we call them unique molecular identifiers, or UMIs, that are basically just little oligonucleotides that bind onto various RNA sites on either the cell or the nuclei. Then we go through a process of amplifying the complementary DNA strands to actually be able to read the amino acid sequences downstream. And then things will be sent off to get sequenced where a machine like the Illumina sequencer will actually read those amino acid sequences to try to identify cogent gene sequences. So the data that we'll get back will be actually counts of or the accurate reads, and then we'll align the data to known references to be able to identify which genes are being expressed in that data. And so that's kind of the very high level process from end to end.
And if I could follow up, Rachel, I think part of one of your like sub questions was like different platforms that can be used for this. So in the workflow that Aaron just described, he mentioned using 10x Genomics as platforms, which is something we use quite frequently here at the Allen Institute. But that is just one technology that can be used for this. So as Aaron described, 10x utilizes like a microfluidic droplet well-based system where you are basically piping in your isolated cells or nuclei and the necessary reagents for reverse transcription. In that they ultimately get packaged in this like little gel emulsion with a particular bead that confers that has the a specific barcode that'll tag that cell. And so everything that happens, all of the initial reverse transcription is sequestered within that little bubble, and that's what allows everything, all the RNA content from that particular cell or nucleus, to be tagged with the same barcode and not be influenced by any of the other barcodes around it. But there are other ways of doing that as well. So there are, instead of using this like gel-based microfluidic system, you can also basically have these cells or nuclei physically isolated in, say, like a 96 or 386 well plate or something, with each well having the necessary barcoded reagents and stuff. And then that plate-based stuff doesn't require as much fancy equipment and technology that 10x does, but ultimately will do the same thing in principle.
Yeah, and then depending on the project that's being done at the Allen Institute, but I mean, 10x, my understanding, is used for the majority of projects, but we still have SmartSeq or some other well-based ones used for some other projects, right? Like PatchSeq is one example where you're specifically extracting out the nuclei from individual cells that you've been recording from, and then that's one example where SmartSeq is used. We do have a live question from one of our attendees. So the question is, does it matter whether you use liquid nitrogen or isopentane to freeze the tissue? How long can you wait to extract the tissue post behavior? I've heard anywhere from under 2 minutes to 60 minutes, and I'm assuming this will affect whether you're looking at nuclear versus cytoplasmic RNA. So there's a couple of questions in there. I can repeat them, but the first one we can start with, liquid nitrogen or isopentane for freezing tissue. Any differences there?
I would say at a high level, I wouldn't assume there'd be huge differences, but definitely using these rapid cooling agents is a must compared to trying to sort of slow freeze the tissue. Really, you start getting a lot of quick degradation as ice starts to form versus, effectively flash freezing. I would say I'm more familiar with using the isopentane approach, especially for primate tissues. And a lot of these are protocols that have been designed by other folks at other universities, such as Doug Rosine at Boston University. He's got a really sort of well-known protocol for freezing the tissue. And as far as the next question of how long to wait to extract the tissue post behavior, obviously the quicker you do it, the better. And a lot of that relates to the integrity of the tissue as you're removing it. For nuclear isolations, we get human tissues that are just, folks unfortunately undergo tragic events, and we get the brain tissues after autopsy. So oftentimes that can be up to 36 hours later. So even 2 minutes to 60 minutes, you probably aren't going to notice huge effects. Granted, with the caveat that you're sort of trying to preserve the tissue as quickly as possible after extraction. With cytoplasmic RNA, so doing single cell versus single nucleus RNA-seq, the tissues are a lot more sensitive. And so that's why we typically use single nucleus RNA-seq, because the sort of the initial steps of tissue breakdown are a lot more amenable to these sort of frozen tissues versus single cell RNA-seq is a lot more difficult to achieve with frozen tissues, I would say.
Yeah, and I'll just say in terms of the mouse work, anytime we're dealing with frozen tissues here, it's flash freezing in liquid nitrogen. I have no experience with the isopentane stuff. And as Aaron said, if at least for mouse work, if we are gonna be flash freezing our brains and then dissecting them out, and then perhaps sub-dissecting out particular regions, we're always going to be targeting nuclei isolations at that point. If we, for any single cell work, we would be not freezing the tissue, for mice at least.
All right, thank you. And that gets into another question, just really, yeah, the differences between single cell versus single nucleus sequencing, when you would use both, when you would use one or the other, and any differences in outcome, how comparable the data is. And one interesting example I think is looking at the whole mouse brain taxonomy from Allen Institute. The majority of that was single cell, but then there were a few brain areas specifically that were single nucleus. So any explanation as to, comparing, those two different techniques within the mouse brain, and also differences, in human and non-human primate as well.
Yeah, I can go. Yeah, so there are a lot of advantages to doing single nucleus sequencing. As Aaron already alluded to, the tissue isolation process, and especially the single cell isolation process, can be taxing on certain cell populations. And so there might be, especially if you're dealing with brain tissue, there might be some small population of neurons that might be lost during that single cell isolation process. And then if that's a particularly rare population, then all of a sudden you won't be seeing that cell type represented in your data. Single nucleus isolation, the nuclei are a bit hardier, and a lot of those cells are less susceptible to loss. And to your point about the whole mouse brain taxonomy, I believe it was probably regions in the midbrain and hindbrain that were mostly suffering from a lot of cell type loss, or a lot of lack of coverage of known cell types. And so that's why they supplemented that with single nucleus data. You do have to be, another nice thing about dealing with single nucleus RNA-seq. If you're utilizing frozen tissue, it's a lot easier to bank tissue for large scale workflows. So you don't have as much of a time sensitive situation as taking fresh tissue and having to do your single cell isolations that day. You can get tissue from collaborators, for instance, or you can get frozen human tissue, for instance. One thing you do need to keep in mind, though, is the difference in kind of the transcript content in the nucleus versus in the cytosol. So when transcription is after everything is, once you're undergoing transcription, then over to translation, the sort of pre-mRNAs that still have their introns intact get spliced out before they get exported from the nucleus. So the nuclear mRNAs that you would be capturing in snRNA-seq will have a lot more intronic content, but that's very easy to get around by just making sure you align your data to a reference that has those sequences in it. You just need to know to do that ahead of time. And also just when you're doing your workflows, like, you always want to be cognizant of RNA degradation and whatnot, but tubes of nuclei hanging out on ice on the benchtop, they can sit there a little bit longer than cells, for instance. And Aaron, I don't know if you want to speak more to the NHP side of things.
Yeah, I mean, the explanation that Emma gave pretty much holds true, I would say, across species. I will say the one difference that at least I've experienced is that single cell RNA-seq tends to give better gene detection, or at least that's how it has historically been. I think there's been a lot of rapid improvements in the quality of single nucleus RNA-seq, so that now we're approaching comparable results, and that's why a lot of the human and non-human primate atlases that we're generating are coming from single nucleus, rather than trying really hard to either maintain fresh tissues or et cetera. But yeah, historically, I think that's why a lot of the early mouse work was done using single cell, was just because of the worry that gene detection wouldn't be good enough using single nucleus RNA-seq.
All right. We have another live question from one of our attendees. The question is, when working with human samples, what is considered to be a good RIN to keep going with the experiment?
So obviously you want a RIN score of 10. But realistically, 10 out of 10 is not going to be the majority of cases. I'll say, we try to keep for a lot of the tissues that eventually become sequence data, we hope that their RIN scores are above 7. That's sort of our sort of lowest passing grade. Occasionally, depending on the experiment, depending on tissue scarcity, all of our tissues are really valuable to us. We may go as low as 6. But really, anything under 6, we start being really concerned about the quality of the tissue and the quality of the sequence data that would be returned. So I would say, shoot for 7 or greater if possible.
All right. Let's see. What are some next questions we can go over? Aaron briefly alluded to this at the beginning, but any species-specific steps or consideration, right? Like we know that the ratio of glial cells is different in our different species, when looking at mice versus non-human primates. And, obviously we have a number of fluorescent reporter lines for sequence that can help for, facts when looking at our mice populations, but we don't have that, right? For other species, for our human and non-human primates. So just some, perhaps considerations or differences with working across, working with different species.
Yeah, I mean, the biggest obvious one is the differences in proportions of glial cells that are present in the data. Um, I think aside from that, there are a lot of sort of primate-specific rare cell types, and when you're sequencing or processing tissue for sequencing, you're collecting sort of centimeter by centimeter chunks. Maybe you go a little bit smaller, but when you're only allowed to load so many nuclei, it's a lot easier to miss these rare types. So certainly, yeah, you have to think about how you're going to slice your tissue and process it to try to maximize the likelihood that you'll capture some of these rare types, thinking about like von Economo cells and other very, very rare cell types where uh, you know luckily for a lot of when you're just doing RNA sequencing and not multiome, which we'll maybe touch on in a little bit, you can actually bank nuclei. And so that's another way in which we benefit um, from using single nucleus RNA-seq for these primate species is that we can try to use sort of standard immunohistochemical antibodies to try to isolate these rare populations and then just bank them over several rounds of prep. And then only after we have sufficient amount to load onto, say, a 10x chip or a 10x port, then we'll actually sequence sort of that pooled set of rare cell types. So I think in the mouse, it's a little bit different where, yeah, because you have a lot of these reporter lines, because the brains are smaller, there's a little bit more of a chance that you'll get sort of the target populations. And in our case, working with these larger primate species, it's a little bit trickier.
Emma, did you have any input for that one? If not, we can move on to another question.
I think Aaron was pretty comprehensive.
All right. Another question is, are there any differences, or perhaps not, in looking at the sparsity of your data? Like, are there certain maybe brain regions or species that you tend to see that more in, or is that something that's just happens across the board?
I'd say in my case, the brainstem is really a tricky spot to try to sequence from, especially trying to enrich for neurons. There's effectively a ton of teeny tiny nuclei that are encapsulated by myelinated tissues. So when you block out any portion of brainstem, you're going to get probably, I want to say 80 to 90% glial cells. And when you're trying to enrich for 70% neuronal populations, you have to go through a lot of tissue to get, neurons. And so I would say, yeah, brain stem's a really tricky one that seems to be unique from other tissues.
And I'd say in mouse, although we get pretty good coverage of all the main brain regions, like the whole mouse brain atlas is quite well annotated. I think, as we alluded to earlier, even that had to be supplemented with single nucleus data specifically for the midbrain and hindbrain regions, and just those particular regions have been a bit messier in terms of being able to cluster the data and effectively get just good coverage and discreteness in terms of the cell types there. And so we've been doing a lot of work to try to profile those regions in a bit finer detail.
Okay, and then moving on to another question that we have live. This question is, I think I might be able to answer this question, but do you have any documentation, notebooks, or published references on how to integrate your spatial transcriptomics data with our in-house single nucleus or single cell data sets, which have been annotated using MapMyCells? So if you've already annotated your data using MapMyCells, you have been given those labels. I'm assuming this is whole mouse brain data, or it could be the whole human brain data, but whatever it is, it has been given the label from the taxonomy that the Allen Institute uses. So then if you were to also look in the whole mouse brain spatial transcriptomic data, if you see subclass or supertype XYZ in your single cell data, you can find that same exact cell type in the spatial transcriptomic data. Because the way that taxonomy was made was that in the whole mouse brain, the gene panel and all these spatial experiments were done based on the data from the single cell taxonomy. And so the same cell type, the same label names are used in both of them. And I can also follow up with this person afterwards, after this talk, to give a more detailed answer about that. But the idea with the annotations is that, yeah, they're in the whole mouse brain, they're the same across those data sets, and same with the Alzheimer's data set. For the whole human brain currently published, we don't have spatial transcriptomic data, but yes, for the whole mouse brain and the Alzheimer's data set, we do have that. And yeah, also if Emma or Aaron, if you guys have any other inputs or thoughts, you can answer this one as well.
Only to say that whole human brain spatial transcriptomics work will be coming in the next couple of years. So yeah.
Yes, there are many exciting projects on the way for both, yeah, mouse and human and non-human primate, and all together, some cross-species stuff that will be exciting to come out in the next few months, next couple of years. So this person, I can follow up with you afterwards. I can email you and show you maybe some of the notebooks or some of the other step-by-step processes we have that can help integrate those data sets together. And yeah. Let's see, let's move on to another question. So we've mentioned, multiome has come up a couple times throughout this talk. So what exactly is multiome? How does it relate to single cell sequencing? And what type of information do you get when you use multiome? And why would you use it? What's it for? And what type of information do you get?
Yeah, I can take that. So I mean, multiome generally is just referring to multiple omics. So like, if you're looking at transcriptomics and some other modality simultaneously, that's going to be kind of multi-omics. So if you're looking at RNA transcripts and protein expression, or RNA transcripts and chromatin interaction, all of that is like multi-omic. But when we specifically talk about multiome, we're talking about 10x Genomics' single nucleus multiome kit, which is specifically looking at RNA-seq and ATAC-seq. So ATAC-seq broadly is a technique that is looking at profiling open chromatin regions. Basically, the enzymes will basically to take, I guess, a step back, your chromatin can be very tightly packed or open up depending on how it's packaged. And open chromatin. Chromatin is typically open when transcription is occurring. And so basically being able to assay open chromatin is a way to not just to see which regions are open during transcription, but also is particularly important for assaying cis regulatory elements. So there are a lot of regions in the genome that are technically non-coding. They don't encode for any particular transcript, but they do play a role. And so there are these elements they're known as enhancers or repressors that can basically help to modify transcription levels of particular genes. Enhancers would increase that, repressors would decrease those. And those elements are very helpful in sort of temporal expression patterns and cell type expression patterns of a variety of genes. And so being able to find a way to examine those in your data is kind of what we typically will use multiome for. And multiome, that's what ATAC will be for, but multiome specifically is also helpful because you're getting RNA-seq and ATAC-seq from the same nucleus. So you can directly correlate gene expression with open chromatin. That doesn't necessarily tell you exactly which regions of when you begin mapping your peaks across the genome from an ATAC space. That does not explicitly tell you what is and isn't an enhancer, but it can whittle down your search space if you find regions of open chromatin that are highly associated with the expression patterns of particular genes. And in my work, in our team's work, we then will take these multiome data sets and identify candidate regions that could serve as enhancers, and will ultimately clone them and test them experimentally to see if they are capable of driving expression in the cell type specific manner that we would predict based on the data. And if those look good, then they can be packaged and utilized as tools for the broader community. And so, yeah, broadly speaking, if the multiome that we do is basically to get open chromatin information alongside transcription detection.
Yeah, I think Emma covered that pretty exhaustively. And I would just say that there's no real trade-off between, well, historically, if you're using 10x multiome and 10x version 3.1 RNA-seq, those are effectively comparable platforms. Now there's new 10x, they call it either version 4 or GEM-X technology that is a little bit more sensitive on the RNA seq side, but there is no multiome component to it. So there is a little bit of a trade-off now between two different platforms of just doing RNA-seq only versus RNA plus attack. But those are considerations to be made when you're designing your experiment.
And I want to piggyback off of something Aaron said, is that also when you're doing the multiome workflow, you're initially profiling things together, but then there's a split, and you're ultimately going to end up with an RNA-seq library and an ATAC-seq library, and you have to sequence those separately. They have different sequencing parameters. So if you want to use the multiome kits to profile your transcriptomes, but you don't necessarily care about the ATAC information right off the bat, you can always just save those ATAC libraries and not sequence them and kind of just bank them for later if then ultimately that open chromatin information is going to be useful. Because we, at least in our experience, have found relative to the pre-version 4 10x kits, the gene detection in the RNA-seq libraries coming from the multiome kits were generally a bit better than the same from just the basic single-nucleus RNA-seq kits. Can't speak for, obviously, the GEM-X until 10x comes out with a new GEM-X multiome type thing, which I assume they're working on. But that's just another thing to think about when designing experiments, is if you want to take advantage of the RNA-seq coming from multiome, but you can at least save a little bit of money in not sequencing those ATAC libraries right off the bat.
All right, we have another live question from one of our attendees. When doing FACS for a single nuclei experiment, are we able to isolate any brain cell type?
Hypothetically, yes. You can isolate any cell type for which you have some sort of reporter for, right? So it's fluorescence activated cell sorting. So you're really just looking at nuclei that are being deflected by lasers of different wavelengths. So as long as you have cells with specific antibodies bound to them whose reporters are, activated by whatever wavelengths you have access to or different lasers you have access to, then in theory, yes. But oftentimes those deflections or the separation of populations isn't always going to be crystal clear. And certainly the more abundant the cell type, the more easier it will be to identify those populations in the plots that are being generated by the software of the fax machine. And so it can be tricky to identify really rare populations. As an example, our team is trying to isolate Purkinje cells, which are, very few and far between in the cerebellum. And when you look at those fax plots, they don't look like Purkinje cells are over here and other cells are over here. It's sort of just an extreme, I guess, edge of the main cluster just happens to be Purkinje cells. And we see in the sequence data that they are consistent with Purkinje cells, with which genes they express as marker genes. But yeah, to answer the question directly, in theory, yes, but it can be tricky depending on, what types of cells you're trying to isolate specifically.
Yeah, I have nothing to add to that. That was great.
Yeah, and that's where we have, all those different fluorescent reporter lines in mice, right, that don't exist, in the other species. That's certainly a factor here as well.
Yeah, and there's a ton of, one of the enhancer screening stuff is also a way to try to identify ways of creating the tools to tag those cell types in the first place so that you could potentially do that. But yeah, depends on what's available or what antibodies you have that you can enrich for particular populations. Easier in mice than in other species.
Yeah, that sort of leads into another question. Yeah, the enhancer screening and the enhancer work that's going on at the Institute. If either of you want to discuss a little bit about that, yeah, why we're doing it, what it could be used for, the genetic tools, any of that.
Yeah, so basically, as I kind of alluded to earlier, we're interested in identifying a number of different novel enhancers that can be used to as genetic tools, basically for identifying particular cell populations within mice, but also, ideally, we'd like to be able to expand that to other species as well, so that researchers can, if you have, some particular, if we're able to particularly tag a fairly rare cell type, then researchers who have an interest in that cell type can go ahead and take advantage of those tools. There's obviously in mouse, we have a lot of transgenic lines as well that also can serve the same purpose. But the sort of reason why we are spending a lot of time screening these enhancers and trying to identify and generate these enhancer-based tools is that maintaining large colonies of mice for transgenic mice can be costly, and it can take time to breed them. Whereas if you have a known, like a very good tool that can target particular cell types, you can simply do a retroorbital injection into a wild type mouse, or into a mouse that has, if it's like a Cree enhancer, you could inject that into a reporter mouse, and that you can, after a couple of weeks, you can then actually see expression and do your experiments there. And so that can also cut down on the costs of like maintaining animal colonies as well, and can be a bit faster as well once we have those tools in hand.
And this also allows potentially for genetic access into some of some of the non-human primate work as well, right? Since we don't have these transgenic lines.
Yeah, that's been a really great advancement in recent years. And actually, at the tail end of my PhD at the University of Washington, I was able to use one of the earlier enhancer viruses that were generated by our genetic tools team to actually infect hippocampal neurons, which all previous attempts using various publicly available viruses were complete failures. So, it was the enhancer technology that really helped to isolate specific populations in my experiments in grad school. So yeah, it's really exciting. And these things are coming online really quickly. They're being applied to, other things such as considering actual, genetic interventions for diseases such as Dravet's uh, syndrome. And so, yeah, stay tuned for more exciting news on that front.
Yeah, and the Allen Institute released the Genetic Tools Atlas a couple months ago, and they recently, in the last, I think, month or so, added just a new batch of AAV enhancers and a whole bunch of tools. A lot of it is towards cell types in the cortex, but also a number in the basal ganglia as well. Those have been some two very big areas of focus. And yeah, someone who had to breed and work with triple transgenic mice for my PhD work, I can definitely vouch that it does take a lot of time and effort. And, these enhancer tools, yeah, make things a lot more straightforward, I think. So it's exciting to see them come out and see how others in the community can use them. All right, so now switching topics a little bit here, we have another question from our audience. And the question is, what is the minimum sequencing depth required in shallow sequencing for map my cells to reliably identify distinct cell identities?
I I'd say I've never stress tested the map my cells algorithm, so I have to say I don't know for sure. Um, Presumably you would be able to get somewhat away with, 20,000 reads per cell, which is the minimum for several of these high throughput platforms. Um, but the issue with that is that a lot of marker genes for these cells are pretty lowly expressed relative to other sort of more ubiquitous, highly expressed genes that aren't necessarily cell type specific. So you probably would lose some degree of fidelity. But yeah, we haven't tried to stress test this, and but perhaps somebody on the computational side uh, would maybe have more inclination. But yeah, I think 20,000 reads minimum is probably in the ballpark of where you need to be.
Yeah, that's what I would guess as well. Yeah, at least, yeah, 20. Yeah, that's what 10x at least recommends. We typically sequence a lot deeper than that, but that's the minimum I would try.
And I do know that if you're using the actual Map My Cells browser tool, it is mapping to the entire mouse brain taxonomy. So if you only have, cells from a certain region of interest, it's assuming the default input is, yeah, that you're going to have marker genes from the entire mouse brain. So that could also affect some mapping. But we also have the code for the mapping from the tool itself on our GitHubs. And if you are familiar with that, it's written in Python. You can actually then subset it a little bit and say, I just want to map to certain brain areas and not do the entire taxonomy. Because it is built to expect, marker genes for the whole mouse brain, because that's how it was built. And I can also follow up, with this person afterwards as well, just to reach out, speak with some of our computational scientists and some of those who actually wrote the code for Map My Cells to, help troubleshoot any issues that you're having. Okay, what are some other questions that we have? How about quality control? Are there certain steps, along the way, like even from just obtaining the tissue, right? I mean, obviously, if you're working with mice in the lab, you're pretty standard in how you're obtaining the tissue. But if you're working with tissue from autopsies, there's a lot more variability there. What are certain like quality control standards that are just present throughout different steps of the protocol?
Yeah, I think we've been alluding to some of these steps in our earlier conversations, but especially with human tissue, we care a lot about the relationship between the postmortem interval, the sort of PMI postmortem interval to brain freezing time, and then looking at the relationship between those factors and then the RNA integrity score, the RIN score. Us and other groups have seen correlations between PMI and RIN score. So, we do these sort of initial observations to get a sense of, will this tissue likely yield good results? And then beyond that, we look at things such as the abundance of nuclei in our FACS populations as we're sorting. We'll look at what the cDNA amplification looks like after loading. And then there's a lot of quality control that goes into like identified doublets in the sequence data and you know actual transcript counts, actual gene counts per cell, and things like that. So at a high level, there are a lot of QC steps along the way. I think I just listed a few of them. Yeah.
And I think it's also important, like a lot of those like post-sequencing QC steps, like doublet detection, gene counts, abnormally high UMI counts, for instance. Other things are good initial filters, even looking at something like mitochondrial ratio, like ratio of reads mapping to mitochondrial genes. But then even once you weed out, if you set certain thresholds there, it's also very good to, once you undergo clustering, for instance, and actually look at your data in that way, there might still be certain groupings of cells that might pass those initial thresholds, but that might still be relative to all the other cells in your data set might be fairly low quality in terms of the distribution of genes that they're expressing and whatnot. And so you can also do some additional QC steps after clustering and kind of go back and see. And oftentimes those kind of like lower quality sort of like junk cells will likely cluster together. And so that can be also a useful way to begin to weed things out afterwards.
Yeah, and you can really see the effects of this quality control, especially in the whole mouse brain taxonomy, right? Because there were close to 8 million cells that were initially profiled, and then only about 4 million of them made it through to the final taxonomy. So the cells that did make it through in that data is extremely high quality and rigorous because of all these different steps, from tissue all the way through analysis, looking at doublets, and you can see it in the final results of the data that it's high quality. All right. So I know we sort of answered this question a little bit in reference to, issues with brainstem and some other areas, but are there any other, particular areas or like parts of the protocol that have just required a lot of troubleshooting, on either of your parts? And I do want to mention also to the audience that all of our protocols are publicly available online from the Allen Institute at protocols.io. So if you have any questions, want to follow through, look through all the different steps, those are also available. Any of the ones that we've discussed today are online.
I could say for the human and non-human primate, we've done a lot of sort of refinement on our nuclear isolation prep, and a lot of that is geared towards the specific step of removing myelin from the tissue. So if you just homogenize your tissue and try to run it through FACS, it's very likely that you'll clog your machine because effectively the myelin or debris from the homogenization will gum up the machine. And so we've gone through a few different rounds and versions of myelin removal, either with using either magnetic beads to attract the myelin, or now we've switched over to using an iodixonol gradient, which effectively filters based off of density, to remove what we assume are nuclei from the more dense myelinated sort of debris. And what we found is that produces significantly higher yields of relevant, in this case, sort of neurons and even, other types of cells relative to debris tissue in looking at these DAPI populations under FACS. But that has taken had taken a long time to sort of perfect, where initially we were having a lot of issues with low yield and actually difficulty during sorts. These sorts were taking a really long time, and now we're getting sort of very clean, very fast sorts, while still achieving the necessary target populations. So yeah, myelin removal is a big one for us.
And I just want to say thank you to all the work that y'all did on those protocols, because I have used that iodixonol-based nucleus isolation protocol when dealing with, we've been undergoing some recent projects where we're trying to profile across the entire mouse brain or across an entire hemisphere at once. And so grinding up an entire hemisphere in a Downs and utilizing that protocol has been very useful, as opposed to some other approaches where we might dissect out a particular region, say primary motor cortex or dorsal raphe or the thalamus, and then with a much smaller tissue input. So yeah, that protocol has been quite good. Some other issues we've had in terms of troubleshooting and haven't really come up with a good answer is there have been some times when we would attempt to utilize transgenic animals and sort for particular neuronal populations, particularly neuromodulatory cell types, so serotonergic neurons, dopaminergic neurons, for instance, and basically isolate enough of those to run like a 10x multiome port, and it's just the survivability there. It was always very difficult to be able to get enough input, and I think that is something that we have kind of had to either pivot away from or we'll focus on troubleshooting at a different time. But that's might be more of a biological issue than a technical issue.
Yeah, and I want to mention that when new protocols are updated or developed, those are added, those are updated as well. I mentioned that they're available at protocols.io, so you can see the different versions of different protocols and see how things have changed over time. So I think that's really helpful for others trying to also do similar experiments. You can just dive right in and see how we're doing it here at the Allen Institute. Okay, so another question, I guess this is more going back towards basics or the foundations, of single cell sequencing. What are some focuses on, certain projects that maybe are involving like short reads versus long reads, or like a 3' versus 5' capture? Like, what are we doing here at the Institute for certain projects? Are we using all those methods? Are we not? When would you use one over the other?
I can talk about the 3' versus 5' aspect. I'm not involved in any 5'. So to take a step back, 10x genomics, for instance, their typical kits, basically the way the RNA is initially captured and then ultimately used to generate these cDNA libraries is there's a poly DT capture sequence on the oligos on those beads, which will bind to the poly A tail that is put onto the RNA transcripts as they are processing. So it's a way to capture specifically polyadenylated mRNA. So that might be pre-splice or post-splice, depending if you're in the nucleus or in the cytosol, but at least you're not just getting a bunch of other random RNA molecules. You're getting things that are going to be serving some kind of function. But that is at the 3' end of the transcript. And so as things are being reverse transcribed and then ultimately getting fragmented down so that you have a kind of uniform size distribution of your library, you are going to be capturing information from the 3' end of the transcript or more biased towards the 3' end of the transcript. If you have super short transcripts, you can probably still get pretty good coverage, but longer transcripts, you'd be missing information towards the 5' end. And there are specific kits that will actually make sure that things are being profiled from the 5' end. So that's particularly useful for immunology work. At the 5' end of the transcript are these sequences that are important for a VDJ recombination that's important for generation of antibody sequences. That being able to capture the 5' end of those transcripts is important for being able to identify differences in those cell types. For a lot of our mouse work, it doesn't necessarily matter to us too much, which is why we typically stick with the typical 10x stuff. But more longer read stuff is also helpful for if you are interested in doing any sort of if you're interested in splice variants or isoform analyses. If you're trying to look at single nucleotide polymorphisms in a bunch of your reads, if your snip of interest is towards the 5' end and if you're just never going to get to it, then you can't even do those types of analyses. So those types of longer read stuff is also useful for that if that's the type of experiment that you're trying to do or the question you're trying to answer. And I don't know if you can speak to any other instances that y'all are using that in terms of NHP space.
Yeah, no, since our objectives are pretty much to identify, the milieu of cell types in these different species brains, we definitely just do 3' and then short reads because we care about trying to get as much sequencing depth as possible. So we just want to see, what are all the various gene transcripts that are expressed to allow us to classify different cell types. So not necessarily interested in transcription start sites or the other facets that Emma alluded to that are the benefits of long read and then 5 prime. Yeah.
Okay, we're almost at the end of our office hour, so we'll just have time for a few more questions. We could, let's see. Some of these protocols that, obviously if you're working with fresh tissue, your timeline is going to vary a lot compared to working with frozen tissue. What are some of the, general start? How long does this process take? Are there any stopping points that you can have?
You go.
Okay. Um, so the initial sort of homogenization to loading step is sort of a day of work, including, tissue breakdown, uh, enrichment or population enrichment, and then actually loading onto the chip. Um, I'd say oftentimes that's the first breakpoint. I think sometimes cDNA amplification might happen towards the end of that day, but sort of the library generation will happen as a separate step, and then sequencing after the libraries are made and banked. Uh, Sequencing will happen for us at least, typically off-site. So tissues will be shipped, and then somebody else will handle the actual sequencing because we're sort of aligned with a bunch of major sequencing cores as part of the different projects that we're a part of. Um, Before in the tissue sort of homogenization steps, if you're just doing RNA-seq, you're not doing multiome, you can bank the tissues or bank the nuclei after either before or after population enrichment. I guess my suggestion would be don't apply antibodies if you're going to bank your tissue. Wait until you're actually going to do cell sorting and then loading. Um, but yeah, that's another breakpoint that a team could hit if they needed to take a pause.
Yeah, and for mouse tissue, we've also found that, in particular, if we're attempting to profile like specific regions of interest, so you flash freeze your brain tissue and you have a frozen brain in a tube, and then you'll cryosection that stuff out. The dissectates themselves can also be stored, and so you can basically decouple like those initial dissections from the downstream workflow, at least in what we found in our mouse tissue. So that's another stopping point. And then you have the RNA-seq workflow. There are a few different. I think they usually spread it over 2 to 3 days, depending on the particular workflow. But once you get through your chip loading, there are a number of different stopping points along the way. So you can it's possible to go from frozen tissue to an RNA-seq library in under 24 hours if you push yourself too hard and don't have a good sense of work-life balance, as I did as a postdoc. But typically, so thinking roughly 3 days, and then sequencing can take time. It depends on how long, especially if you're working with an external core, like what the demand is. And then once you get data back, then there's also, depending on how much you have, then there's also alignment of everything, depending on how much compute power you have. So, probably good estimates, probably from from tissue isolation to like having data in hand that you can analyze, probably a couple weeks, if nothing horrible goes on in the process.
All right, we have one more live question from the audience. The question is, can you give recommendations for lab protocols of spatial transcriptomics? How do you do it in human samples when sometimes it is difficult to exactly match the same anatomy?
So I'm not on the spatial transcriptomics team, so I'm not involved in the bulk of the work that's being done there for the HMBA atlases. I do know that there's some element of light treatment that happens prior to actually mounting on the slides that will be sent off for the different platforms, whether it's Visium, um, or MFish, or sorry, Merscope. But uh, outside of that, with respect to how you would do it in human samples when the anatomy is difficult to align to, um, I think that becomes sort of a judgment call of can you make sense of what you're mounting to, and also, it shouldn't affect anything to try to take some uh, you know, top block face sections of the tissue and try to do just a standard, even just a Nissl stain to see what that anatomy looks like under the microscope. And yeah, I think it can be tricky depending on where the tissues are being sourced from to know that, you know, I received the head of the caudate or I received the body of the caudate, and knowing which orientation should be which. I know that's not always the clearest, but hopefully that wouldn't affect the transcripts that you're getting. And if anything, you know, because you are taking thin sections off the face of the tissue, you'd hope that the transcripts that are being read out somewhat align with known references, like even the uh, to be completed Allen human transcriptomic brain atlases. So yeah.
Yeah, as Aaron mentioned earlier, we do have a lot of current projects underway at the Institute looking at, yeah, spatial transcriptomic, especially in human and non-human primates. So hopefully once those are out soon, that can help answer some questions. And we did have last fall, we had a live webinar from our spatial transcriptomics team. It's on the YouTube channel. So if you just search Allen Institute spatial transcriptomics on YouTube, that should pop up. And we had different members of our spatial transcriptomics pipeline participate in that webinar. So hopefully that could also, provide some answers. But that is all the time that we have for today. Thank you so much again, Emma and Aaron, for joining us and for answering all these questions. And thank you for all of our attendees. And for a few of the attendees who answered some questions that might require some follow-up, I can email you afterwards and we can work together and troubleshoot some things. All right. So thank you everyone for joining us, and I hope you have a good day.
Thank
Thank
you.
you.